Let's get started. So welcome and thank you for everyone for joining to the next talk in the CCI seminar series for the spring and summer term. As usual, can I ask you to have your microphones muted and your cameras off until the question session so as not to disturb anyone. I'm absolutely delighted to introduce Dr. Maria Dornelas from the University of St Andrews. Dr. Dornelas is a reader based in the Centre for Biological Diversity within the School of Biology and holds a PhD from James Cook University. Dr. Dornelas' research focuses on quantifying biodiversity and understanding the processes that shape it, often focusing on tropical systems and specifically coral reefs, but also with work in tropical freshwater fish and mangroves. Dr. Dornelas' research combines ecological theory, synthesis of existing data, and fieldwork to answer questions with within the disciplines of community ecology, macroecology, and biogeography. If you have any questions, please keep them in mind till the end of the presentation. And at that point, feel free to put them in the chat or raise your hand directly. And I'm really delighted that today Dr. Dornelas is going to talk to us on the topic of nuance in biodiversity patterns. So at that point, I will hand it over. Thank you very much.
Hello, everyone, and thank you very much for the invitation to give this talk. I am going to share my slides now, and hopefully you can all see them. Please let me know if not. Yeah, okay, right. So the title of my talk is Nuance in Biodiversity Change. And what I hope to show you today is that biodiversity change is incredibly variable, and it depends on a number of things, including but not only on scale, spatial scale, in particular on the geography. So different parts of the world are changing in very different ways. It is affected by a number of different drivers of change with often sort of conflicting effects on biodiversity change. And then, like, sort of my main take-home message is that actually all of this variation really matters, and it matters for a number of reasons, including not just our understanding of how the world is changing, but also what we do about it. Okay, so the work that I'm going to be presenting here today is pretty much all of it based on the Biotime database, which is a database of biodiversity time series that's publicly available for people to use, that combines the work of about 300 biodiversity researchers who have been sort of keeping track of the abundances and identities of individuals of all kinds of different taxa and different ecosystems throughout the world. Oops, I don't know why. Oh, here we go. The database is still growing, but it currently has almost 13 million records spread throughout the world, over 600,000 locations, almost 50,000 species we're keeping track of, and it currently includes 443 studies, although because it's a growing database, this number is probably out of date. And it's time series, really, so it spreads from the late 1800s to the present, although the vast majority of the data is from the past 50 years or so. I don't know why it's not one. Okay, here we go. So the first study that we published on this database was back in 2014, when the database was about a fourth of its current size. And at the time, we had a look at how a number of different diversity metrics, but for the purposes of this talk, I'm going to focus on species richness on the y-axis here, which I've logged just for the purposes of plotting it, how species richness changed as a function of time on the x-axis here. And we fitted a hierarchical mixed model to these data, keeping all the studies separate and estimating the trends within each study, where a study is defined by the methodology that was used to collect the time series. And what we found was that contrary to what we were expecting, so we came into this thinking that we were going to quantify differences among taxa, etc., in biodiversity declines. What we found instead was that, sure, sometimes in some time series we could find declines, but there were roughly an equal number of time series where we found actually increases in species richness, as well as a great deal of them where we couldn't detect any change. And on average, that's the black line that we see here, actually these gains and losses were balancing each other, so we couldn't actually detect a change in species richness because of this balance in gains and losses. We also looked at compositional change measured with the Jacquard similarity for this particular plot, but we measured it in lots of different ways. And Jacquard is a compositional similarity metric that goes from 0 when the two samples that we're comparing don't have any species in common, to 1 when every species is represented in the two samples. And we compared each point in the time series with the start of that time series. And what we found was that, contrary to what we see in species richness, there was a pretty consistent decline in similarity. And what this means is that we can see, and this is true like for the overwhelming majority of the time series, as well as on average. And what this means is that we have a consistent turnover, a consistent change in the composition of assemblages pretty much all of the time series. And so out of these 202 lines that I'm showing you here, there is one that was somewhat controversial. It's the black line on the left, because it kind of flies in the face to what we expect and what we think is happening on the planet. So we don't see declines in species richness. And what we've been trying to do ever since is to try and make sense of that result, to try and understand how it is that that is possible, given what we know about the ways in which humans are transforming the planet. And I hope to persuade you that to understand that result, we really need to unpick what is happening behind all of this variation in trends that we see on the left here, as well as keep in sight this changing similarity. Okay, so the first element that I would like to focus on is the element of scale, and spatial scale in particular. So one of the reasons why we felt we were so surprised by not finding declines in species richness is because of what we know about what is happening to extinction rates. And there's multiple lines of evidence that suggest that extinction rates are highly elevated, up to orders of magnitude more than under normal conditions. And so how is it possible that we're losing so many species and yet we don't detect declines on average in species richness? And in trying to make sense of that, Brian McGill, Nicatelli, and McGren and I put forward a paper in 2015 that argued that there are enough species and enough individuals on the planet that actually change at different scales doesn't need to be in the same direction. And what I mean by this is that it is possible to be losing species at the global scale, so these elevated extinction rates that we have, and through the redistribution of species on the planet, not necessarily be losing them everywhere at local scales, which is what we were looking at in these data. And so we put this forward as a hypothesis, and then we went on to test it with data. So this is part of a working group, an SD working group called S-Change that I co-led with Sarah Supp and Maria O'Connor, and this particular output was led by John Chase. So we assembled time series that varied in the spatial extent, and we looked at the log ratio of species richness in these time series between the start and the end of the time series. And so that's what we have on the y-axis here. And one the number is positive, then we've had a gain in species richness. When that number is negative, we've had a loss in species richness. So let me show you what the data also looks like. So this uses a biotime data set. Largely the white circles here come out of biotime, but it was supplemented with some other data as well. But because we wanted this to maximize our scale axis, we also included additional data coming out of checklists, because it turns out that we use different methods to study biodiversity change at different scales. And because we use different methods to study biodiversity change at different scales, it's actually remarkably difficult to know for certain that the change we see with spatial scale is caused by spatial scale rather than the method involved. But certainly we detected some change in the direction of biodiversity change with spatial scale, and most certainly we detect much more variability at local scales than we do at the global scale over here. Right. So the second item that I want to focus on is geography. So we hypothesized that a lot of the variation that we saw in that plot I showed you on the left was actually driven by different parts of the world undergoing different changing in different directions. And again, this is another S-Change working group output, and this one was led by Shane Blows and Sarah Sapp. So what we did is, again, using the biotime database, we fitted models that try to unpick this geographic variation in biodiversity change. We call this the geography of biodiversity change. We fit the different models. This one that I'm showing you in particular estimated rates of change for different biomes of the world. And just to help you interpret this map, so here we have this black dot, and the arrow bars around it represent the global mean. And somewhat reassuringly, despite the database having tripled by the time we did this analysis, that result didn't change. So it's still indistinguishable from zero on average and across the planet. And then the colors that you see on the map are the different biomes, the different marine biomes for this map in particular, that have been color-coded according to whether they're above the global average in blue or below the global average in the warm colors. And what I kind of see in this map is that there is definitely some geographical variation here. And most, but not all, of the warmer colors are towards the center of the map, and most, but not all, of the blues are towards the tops of the maps. And so I would say that perhaps this suggests to us that there is a signal here of polewards migrations of species ranges. So as the world warms up, we're seeing sort of range expansions towards the poles, and declines in diversity in tropical regions. And there's definitely independent evidence to suggest that that is the case. Oh, that's just the arrow to help me point that out. And this is a similar map to the one I showed you before, but for the terrestrial rather than the marine biomes. And here, the story, I think, is slightly more complicated. I don't see a very clear pattern. There is definitely spatial variation, but I don't, in that some biomes seem to be changing in one direction and other biomes in different directions, but the patterns are not as clear. And I don't see an overall pattern that arises out of this. We looked at compositional change as well. And I'm plotting the marine and the terrestrial biomes side by side, because I want to point out that in terms of compositional change, an important point to come out of this was that the marine realm seems to be changing in composition a lot faster than the terrestrial realm, and you can see that because most of the marine biomes are color-coded blue, which is again above the global average, and most of the terrestrial biomes are color-coded red, which is below the global average. And again, the global average for turnover, so for compositional change, is significantly positive. So there is a systematic change in composition taking place in this expanded data set. And so far I've been talking to you about a pretty sort of big picture, broad stroke image of what's going on. And what I want to do next is zoom in on a particular part of the planet where I've been working for many years now, and kind of go look under the hood in one of the time series that contributes to biotime. And that's specifically Lizard Island, where I've been working since my PhD. And here I've just sort of pointing where Sydney is, because that's going to be relevant in a couple of slides from now. And so that's Lizard Island here, and over here is Chamorro Reef, where I've been working since my PhD, and fortunately I can't travel there now. And in the last few years, we started using underwater drones as well as swimmers to create 3D maps of the reef. And you can start, like, as we zoom in further, you can start to see the branching corals and the massive corals, etc. in there. Here they are in color. And we've been keeping track of how this particular reef has been changing through time. And we do that by using these maps and then annotating them with the identities of each of these coral colonies that you see here. Or at least we were in the middle of annotating this when we were interrupted. So this particular image is from 2014, and at this point, for this particular section of the reef, we had about 40% coral cover, which is the average for a healthy reef, or was the average for a healthy reef in this part of the world at the time. And and within a section sort of roughly the size of a rugby pitch, we counted 110 coral species. Now, as I said, we were interrupted in the process of data collection because there was a cyclone, and so we had to be evacuated. And it actually turned out to be one of the most extreme sequences of disturbances that, I don't know, definitely for this reef on record, in that there were 2 category 4 cyclones that hit the island from 2 perpendicular directions. And then that was 2014 and 2015, and then 2016 and 2017, there were extreme marine heat waves, which resulted in bleaching. And for those of you who are maybe not coral reef biologists and may not know what bleaching entails, the corals get most of their energy from symbiotic algae. And when the water gets too hot, they expel it, and if bleaching is prolonged, often they die. For this reef in particular, the combined effects of all of these disturbances were stark. We had over 90% mortality. We have a 60% decline in species richness, because there are, it was not more than that, because if you look very closely, there are some little bits of corals here and there that haven't died. And we went from 40% coral cover to less than 2% coral cover. So a very extreme decline. So very different scenario. So that was one example of one of the time series that's in biotime, which leads me, because I've been talking about extreme climate change associated effects to the topic of drivers of biodiversity change. Now, we now know, we have for a number of years, that there's a number of different drivers of biodiversity change that we recognize, so climate change being one of them, human use and it's, I guess especially overexploitation being another one, just the presence of humans and urbanization being another, pollution and invasive species. All of these contribute in different ways to biodiversity change, but they're not randomly distributed on the planet, right? So they are distributed in specific ways, and this creates a mosaic of different intensities of cumulative change on a planet. That's what we have on top here, as well as different combinations of different drivers. So if you look at the maps on the bottom here, you will see that different parts of the planet are exposed to different cocktails of drivers of change. And in a paper that was led by Diana Bowler, we quantify these and we call them anthropogenic threat complexes, and we kind of map where in the world you find these different combinations. So it's a pretty complex mosaic, actually, which I think goes some way into explaining some of the geographic patterns that we saw before. So and the other important point to make is that there is definitely spatial autocorrelation amongst these drivers in that often they're associated to the presence of humans, and that is true out of pretty much all of the drivers except climate change. So climate change is most intense in some parts of the world where we don't have many humans living at all. So it's the most orthogonal of the drivers of change, and this is especially true in the terrestrial realm, and less so in the marine realm. So in the marine realm, we actually have intense warming in some parts of the world where we also have lots of humans. Some of the work that we've been doing since is sort of putting these two things together. So we have these drivers of biodiversity change, we have a biodiversity time series, how much of the former explains the latter? And Laura and Town led a paper where we matched the biodiversity time series that we had with temperature time series, and then we quantified a change in temperature in these matched time series. And what we found was that, and this is like totally not surprising, is that on average the time series detected warming, although some places were cooling as in the specific time frames of the biodiversity time series that we had, but on average they were warming. And so we also this is to remind me to tell you that because we were focused on time series with a minimum of 5 years, we ended up not having enough data from polar regions and tropical regions, and so we focused this analysis on temperate regions only, and this gave us roughly 21,000 time series to play with. And these are the matching species richness trends, and what we asked was whether or not the trends at the top, the trends in temperature, explain the trends in species richness. And what Laura found is somewhat complicated. So here we go. So in the marine realm, we see that the warmest places have gained species on average with increasing temperature. So on the x-axis here, we have temperature change, so positive on the right and negative on the left, so warming versus cooling. And then on the y-axis, we have the long-term temperature, so warmer on the top, colder on the bottom. And where we on the background is color-coded according to whether species richness is increasing in red or decreasing in blue. And what we found was an effect that shows warmer places that have warmed have on average gained species, and colder places or places that are cooling have on average lost species. And this is for the marine realm. For the terrestrial realm, we couldn't really detect an effect at all. We have a lot less data for a terrestrial realm than the marine realm, so I'll just leave it at that. So if this is surprising you, given that I've just gave an example of massive losses of richness caused by warming, I want to point out again that this is temperate data only. And if we have migrations of species towards the poles, we would actually potentially expect to have the diversity of tropical regions extending into the temperate regions. So this is potentially what we are detecting here. Just to give you a concrete example of that, I showed you before where Sydney was relative to Lizard Island. And here, these sort of blobs on a rock are corals that we can now see in Sydney Harbour. This is a photo taken by my colleague Josh Maiden a couple of years ago. So they were not there 10 years ago. So we're definitely beginning to see corals colonizing Sydney Harbour, very clearly not in the tropics. Just in case, to make it clear, the previous photo, this is not a coral reef, right? This is what a coral reef looks like. So just because we have gains in some places and losses in others, the gains and the losses do not necessarily mean the same thing. And so we need to be very conscious that we don't just look at the balance, we don't just look at the net, but we actually look at what is gained and what is lost. Because these are not necessarily equivalent. Right, so the gains in some places are not equal to the losses in others. Right, so the next example that I want to talk about goes back on land and looks at the effects of forest cover change on biodiversity change. So for this is work that was led by Gergana Daskalova, a PhD student and co-supervised with Isla Maya Smith, and that uses not just Biotime but also the Living Planet database. And Gergana, like similarly to what Laura did before, Gergana matched the time series in the Living Planet database and in Biotime with time series of forest loss, estimated from land use change, spatial layers. And what she found was that population trends following peak forest loss were amplified, but it was not just the losses it was not just the declines in populations that were steeper, the increases in populations were also steeper. So this is coming out of the Living Planet database. So there are both more increases as well as more declines in abundance coming out of that analysis. And in terms of species richness, the story is slightly different in that we see declines amplifying, and although we see lots of increases, they are not steeper than they were before before peak forest loss occurred. Right, so I showed a great deal of variation in space, in scale, variation caused by different drivers. And the last point I want to make is that all of this variation really matters. And I'm going to illustrate that with another study that was led by Brian Long. So Brian, again, using the Living Planet database, went and looking under the hood of the data that are used to calculate the Living Planet Index, which you may be familiar with, is often every couple of years a Living Planet report comes out, and this is the headline number that comes out of that, showing that we have in excess of 50% decline. This exact number depends on exactly what data is used, as well as what weighting factors are put in there, but it's always more than 50% decline in the geometric mean of populations. What Brian Lung did was to try and look at what is the variation behind this number, and specifically how sensitive is it to extreme declines within the database. And what we found was that removing as little as 2.4% of the most extreme declines actually reverses this trend. Rather than having the vast majority of the populations declining, as one might assume from just looking at the one line, what we have instead is a huge variation in terms of population trends. There are some very extreme declines, there are also but fewer extreme increases. And then there is a huge majority of populations that actually are not changing that much. And so what Brian and the rest of us argue in this paper is that rather than focusing just on the mean, we need to look under the hood and look at the variation that's in there. And it might be, it's probably beneficial to use this variation to help us prioritize. And this is also matched with a similar pattern that we see in the biotime time series, where we have small proportion of species that have either colonized or gone extinct, and then a huge majority, so 92% of the populations that are always there, and those are roughly balanced in terms of the ones that are increasing and the ones that are decreasing. So we shouldn't lose track of all of these just because we care about these ones, and we should actually, if we particularly care about the things that are going extinct, we should be focusing on those rather than assuming that everything is undergoing the same trends. And just to give you a concrete example of what I mean by these sort of declines, this variation in population trends. I'm originally from Portugal, and when I started my undergraduate degree, I remember talking, the example of the Iberian lynx was taught quite a lot in my undergraduate degree as an example of a species that was pretty much probably condemned to extinction. It was incredibly rare. By the time I started my PhD, there were 94 of these beautiful cats left in the wild. Last year, there were 855. Now, I'm not saying that, you know, that's it, we're done, awesome, it's out of the woods. It's still a highly endangered species, but surely this is a huge increase, and we need to be able to celebrate these nothing short of victories, and that not only has this species not gone extinct, it has massively increased in the space of 20 years. As part of our discourse when we talk about biodiversity change. And so that's it. So, you know, my final slide is just to say that whenever we talk about whether or not biodiversity is declining, the take-home message is that there is definitely a great deal of things happening. There is a great deal of biodiversity change happening with replacements of species and compositional change, but there are both gains and losses happening at the moment. And I think these gains and losses should be used to help us set priorities, both in identifying the places that need the most urgent attention. And they're also an important reason why we need biodiversity monitoring to become less biased than it currently is. And that's it. Thank you.
Okay, thank you for an absolutely fascinating talk. I'm sure there'll be lots of questions. In terms of questions, feel free to turn your cameras on now if you want to. If you have a question, either raise your hand, turn your camera on and wave, or place it in the chat. Does anyone want to get us started? Okay, I have one from Benno Simmons. In the geographical analysis, do you think we can trust the trend estimates in different regions and their directions relative to the global average? Presumably the time series in each region are non-random and incomplete samples of the populations in each region and globally.
Okay, so I think the honest answer is that it depends. For some parts of the world, yes, absolutely, we have enough monitoring that, well, I guess I should say for some parts of the world and some taxa, we probably have enough monitoring to have a reasonably good idea of what's going on. For other parts of the world, absolutely not. And it's true that even though we color-coded the biomes for everywhere where we had some data, for some of those biomes, we had a lot more data than others. And so I guess what I would say is that this is very much a macroecological analysis, which means we trust the broad structure, but not the detail. If we really want detail, then we need to do more, enough data that we can say things with a decent degree of certainty. Does that answer? Terms bias, absolutely, yes, there's a lot of biases. And what we can do for the time being is I guess, be open and transparent about these biases and iteratively work towards reducing them. I was a little bit concerned I was going over time, and so I didn't actually do my plug about getting more data. But if you have data that you want to contribute to, Biotime is very much a growing database. Please feel free to get in touch and help make it less biased.
Are you targeting any particular ecosystems or regions as the key ones that you need to kind of help fill these kind of global and regional trends?
Yeah, I mean, absolutely anywhere but Europe and North America, if I'm honest, because that's where we have most data. So Africa, Asia, the deep sea, polar regions, taxa that are not birds or plants. These are all things that we have, and and I love all of these things, and I don't want to, but these are all of the things that are like grossly underrepresented, not just in Biotime, but generally in biodiversity databases. So all of those are relevant.
Great. We have a question from Phil Martin.
Okay. Hey, Maria, that was really interesting. Thanks. So my question is regarding the stuff to do with the Living Planet Index analysis. And we looked at this in Journal Club. We looked at that paper a few months back. So some analysis by people at ZSL suggests that if you remove the most strongly increasing populations from the LPI dataset as well, you still see an overall decrease in the Living Planet Index since the 1970s. And presumably this means that the LPI is very sensitive to outliers, and maybe being a geometric mean, you might expect that. Do you think that changes the interpretation of what you say in your paper, or do you think what you've said in your paper still stands up?
Yeah.
Or what kind of added nuance do you think it adds?
Okay, so there are 2 points. So 1 point, I don't think we imply anywhere in the paper that there aren't declines, or even that there aren't more declines than increases in populations within the database. There's another view in the paper that makes that exact point. That was not the point of the analysis. The point of that analysis was just to show that the index is very sensitive to outliers, and so we need to take it with an extreme a pinch of salt, and maybe think about, well, complementing it, if nothing else, with other analysis is what I would take from there. Yes, it's true that if you just remove the positives, the index also changes. It becomes more negative, although not as fast, although we might explain that statistically. And there's also there are statistical reasons which I won't be able to fully reproduce now, but to get in touch with I'll get my head around it. That, like, if you do it paired, there are statistical reasons for why you would expect it to not change very much. Does that kind of answer your question?
Yeah, that's great. Thanks very much.
As a follow-up to that, is one of the challenges with the LPI is that it's quite a compelling metric. So one number that describes kind of changes on a planetary scale, and maybe describing those nuances is often quite a hard task for communication and scientists.
Yeah, that's a really good question. In that, like, it's always a tough balance to strike between being simple enough that everyone can follow what you're saying, and not so simple that you're kind of misrepresenting what's going on. And I think that is a consistent, it's a continuous balance that we need to seek. And I guess the point of my talk, the point that I'm trying to make in this talk, is that I feel that when we talk about biodiversity, we almost always only ever talk about the losses. We almost never talk about the gains as well. And I think that actually misrepresents what's going on. And I think we need to be a little bit more nuanced in how we talk about biodiversity, and how it's changing. But yes, I totally recognize it. Like, whenever you say, "Oh, it's complicated," you've lost half the audience, and that's tricky because getting the audience on board is a big part of what we're doing. Yeah. I also think that sometimes we underestimate our audience, and we do to make it too simple. Yeah. I'm not sure if I answered.
That's great, thank you. So I'm going to skip to the next question from Diego. How often is the Biotime database updated, or how sensitive is it to detect changes in places where there is good data?
So the first paper was published, the first paper making it public was published in 2018. We're now gearing up to release version 2.0 of the database. So it's a really good time to get in touch, plug again, if you want to contribute data. I don't think we have the resources to update it more frequently than we currently are. And bear in mind that this is a legacy database from an ERC project where I was a postdoc, that we have sort of kept going out of getting bits of money here and there. And so this is the best that we can do at the moment. We're not, like, there isn't a whole institute with a team of people behind the database. But it is growing, and one of the things that we do, one of the, for me, important things of the database is allowing the people who collect these data to get credit for the data. And so that's why I released all data contributors who want to be listed as co-authors on the data paper, and so they can be cited whenever the database is used. Yeah.
That's great. Thank you, Maria. Thank you.
Thank you.
Thank you, Diego. Do we have any other questions? Okay, I had one related to the trends in the marine species. It's kind of an area I'm very much interested in. So you found some trends within the temperate marine species relative to temperature. Do you think, with kind of your work on kind of coral reefs, that we would see the opposite trends in tropical regions when species are being pushed towards maybe their thermal limits or in concert with other kind of human pressures?
That is definitely my hypothesis, and one of the reasons why I was keen to so I think we actually had contrasting hypotheses for what was going on in the temperate regions versus tropical regions, and that's why I was keen to only do the tropics if you had enough data that I was confident that we were representing it accurately. I do think that what we know about what's going on, there is a reasonable chance that we would detect different patterns. There's independent lines of evidence, including work by Mark Costello, where they are beginning to detect bimodal latitudinal gradients of diversity. So a dip in the tropics where we have higher temperatures and temperatures. So that is a hypothesis. I would love to be able to test that, but we just don't have enough data, and especially not enough long-term data from the tropics to do that, which is a real shame.
So I have a question in the chat from Ian Burfield. Do you have any suggestions for how we can convince governments and others to invest more resources and be more representative in biodiversity monitoring?
Well, I definitely think it shouldn't be coming out of the science budget. I think it needs to be independent of the science budget so that we don't feel like it is competing with the normal science that we do. So monitoring, and I guess monitoring to inform policy in particular should have an independent budget. I'm actually currently involved in a European project called Europabon that's tasked with designing a more representative biodiversity monitoring system for Europe. Now, unfortunately, I actually think that Europe has a reasonably okay biodiversity monitoring system as it is. So what we need is an Europabon, but for the planet, so that we actually start to represent better the parts of the planet where we don't have wealthy nations and we have the vast majority of biodiversity. How do we do that? I don't know. I think at some point ecologists are going to have to do what astronomers did when they wanted the Hubble telescope, and just everyone needs to join in together and say, like, we really, really need this. This is the piece of kit that everyone needs. And it's not instead of normal research, it's in addition to, yeah. But I guess first, like within the field, we need to agree that this is what we need. I think there's also a question in the chat from Claire. Hi, Claire. I haven't seen you in a while. Asking about what we're doing next. Okay, lots is the answer. We have one analysis that we're almost done with, looking at spatial compositional change, so changes in beta diversity, because one of the hypotheses that we had is that, oh, okay, so maybe we don't detect losses at the local scale, but we have the same winners everywhere and different losers in each place. So can we detect that when we look at patterns of beta diversity? So that one is almost done, and again, that's work that's led by Shane Blouse and includes John Chase and Brian McGill and other people. We also have just started a working group called Estetra, where we are matching biodiversity time series with species traits. And we're starting to look at, what are the characteristics of the winners and the losers and that we have. So can we sort of dig deeper and actually have more information about what does this change entail? What are the implications of this super prevalent turnover, for example? So, like when you have replacing things that are not replaceable, and yeah, but that is a challenge to do well, I think. So, but watch this space.
Thank you. Are there any more questions from the audience? Okay, well, with that, I would once again like to thank Dr. Dornelas for a really fascinating talk that covered lots of really interesting topics. Just to remind people, there is another CCI seminar in 2 weeks, which is Robin Chasden speaking on natural forest regeneration. And with that, I'd just like to forward my thanks to Dr. Dornelas for joining us today, and also for all of you for joining us. Thank you very much.